All right, it's time for Chapter 18, where we are going to focus on the geography of evolution. And here I've started with an image of both an emu and an ostrich because they hit upon this topic that we've already spoken about, the geographic component of biological diversity, because you're aware that both of these are large flightless birds that are grouped into um, the clade that is referred to commonly as ratites. So, ratites are a very interesting group of birds. There are a number of different species, but it's not so much the fact that they can't fly or that they are so large that we're interested in today, it's where they occur. Okay, so what we're going to be thinking about in this chapter are where organisms are and why they are there. Okay. And in this, I'm going to continue talking about ratites here for a moment. And as you know, ratites are a large group of flightless birds that exist in the areas highlighted in red on the map on the right. The names on the continents are incorrect. Ostriches do not live in South America, they're mixed up. I don't know who made this. They're good at graphics, not great at biology or biogeography, that is. But what I want you to realize is that this is an interesting pattern biogeographically. Why is it interesting? Well, if this were just any old bird group that was distributed across these continents, wouldn't be so surprising. The fact that these are 100-pound or multi-100-pound flightless birds that don't swim, that tells me something. Something's interesting here. And from a biogeographic perspective, it's interesting because there's got to be some explanation for how these birds came to be on these very isolated land masses. And if we think about it, we don't think about biology in a vacuum. We think about it in the context of all other aspects of the landscape upon which these organisms reside. And when we think about it that way, we go back in time because ratites are an ancient lineage, and we think about, were these continents always in this same position? And indeed, they were not. They existed in what was, as parts of what was once a large supercontinent of Gondwana. And you can see here on the map on the left that Gondwana consisted of South America, Africa, India, Antarctica, and Australia. Okay. And with these continents all together in one giant landmass, it should be clear to you that an ancient lineage like ostriches, or indeed one like crocodilians, another one that we're not talking about in this chapter but has a similar distribution, these are species that are very old, or lineages that are very old, and certainly existed at the time when these continents were in this position, and rode the continents as they drifted apart and moved to their current positions. So the differentiation of those populations that live on these different land masses should be attributed to the process that we know as vicariance rather than dispersal. Clearly, these ratites are not dispersing across the Atlantic Ocean. Now, as we start digging into the geographic patterns of biodiversity, I want you to know that you know more about this than you probably think. Okay? You have some understanding of biogeographic patterns already just from the random things that you've seen and read. Okay? And I want to make that clear in the example I'm going to show you on the next slide, where if you knew that Dr. Noonan was going on a trip this summer and posted on his Instagram some photos that he took on a hike one day, and I said, hey, today I was walking around southern Brazil in the forest, and I took some really cool pictures of the wildlife I saw walking around the forest. And here they are. Look at these amazing animals that I saw walking around while I was on my trip in southern Brazil yesterday. You are going to call, and rightly so, bullshit on the fact that I took these pictures walking around Brazil. Okay? Because clearly you know that you must know at least 3 of these things don't occur in Brazil. Okay? And in fact, none of these occurs in Brazil. So that we've got an Asian species in the tiger, we've got a North American species in the moose, an African species in the elephant, and an Australian species in the kangaroo, right? And the fact that you have this basic understanding of biogeography speaks volumes to the true clarity with which you can recognize and identify certain organisms as being characteristic of certain regions of the globe. And the fact that all species are not everywhere and uniformly distributed is one of the things that really spoke to Darwin and Wallace as they developed their theory of evolution. Alfred Russel Wallace, remember, he was one of the co-fathers of the theory of evolution. He's the father of the field of biogeography. He came up with this idea that these biotic realms are biologically real. So these geographic regions can be ascribed some characteristic fauna. So the Australian realm, for example, is characterized by a great diversity of marsupials and a dearth of other species of mammal that you might find in other areas, such as North America or Africa. Neotropical regions have lots of things that the Nearctic region, which is North America, does not have. So things like monkeys or a number of different amphibian groups, mammal groups. There are a lot of characteristic species in the Neotropical region that you don't find in Africa or North America. Okay, so each of these regions has a very characteristic fauna that allowed Wallace to describe and delimit these realms. You'll notice there are a few bands where there's some interaction, but say between the Palearctic and the Oriental region, or the Ethiopian and the Palearctic region. But it was because Wallace paid such great attention to the patterns of the species that he collected that he was able to make this classification system. And you'll note somewhere between the Australian and Oriental region, there's a little red dashed line there, Wallace's line. And this is actually part of, or one of his most famous discoveries, in that this line delimits a region in which land masses to the north and west of this line have primarily oriental fauna, so things like rhinoceros and tigers, whereas things to the south and east of this line have an Australian fauna, so things like marsupials and Australian species of bird and reptile. And this is because there's a very deep channel there that physically separates them, and those organisms have never had the opportunity, despite changing sea levels over time, to walk from one of those places to the other, walk across that line. Whereas on either side of that line, those things that are now islands have been connected in the past, allowing Australian organisms to move around all of those pink islands and Oriental organisms to move around all of those orange islands. Okay. So, what about all of these things that we see on different continents that inhabit similar environments and have a similar appearance? Well, you'll remember we touched on this very specific situation in the past when I spoke to you about the principle of biological convergence. So different lineages converging on the same phenotype and the same ecology because they are exposed to similar environments. So in this figure are both cacti from the Americas, the deserts of the Americas, and euphorbs from the arid regions of Africa. Okay, so don't be confused by these similarities on very different land masses, but they can confuse biologists who are attempting to interpret biogeographic patterns. Now, does that mean if we observe very similar organisms in very different areas that it clearly must be convergence? Well, the ratite example suggests no, it should not always be thought to be convergence. So, how do we differentiate these? Here's a great example. Okay, there are two species of alligator in the world, one of which you're probably relatively familiar with because you live near or at least during the times when you're in Oxford, in an area that is inhabited by one of these species, Alligator mississippiensis. Okay, this is one of two species in the world. The other is the Chinese alligator, this fellow on the bottom left, who's found in a very small region of China, just west of Shanghai in the Yangtze River. And they are a much smaller species, approaching 5 foot in length. They have a much shorter head. They're clearly different, but they are clearly not crocodiles. They are alligators. And so this very separated distribution is interesting because it takes a while for for biologists to document this and then reconstruct the evolutionary history to determine, are these two simply convergent lineages, or are they in fact monophyletic? And this is an interesting biogeographic pattern that deserves exploration in order to determine the explanation for this pattern. So what we've seen here among the geographic ranges of alligators, and illustrated here in the species of burrowing tortoises of the genus Gophurus in the United States, are what we call disjunct distributions. Okay, now you might look at this and think you've already learned the term for this, allopatry. And it's true. Allopatry refers to two populations that are isolated from one another, and that's a mechanism of speciation that you've learned. Once isolated, these populations evolve independently and become different species. That is, in fact, allopatric speciation. The populations exist in allopatry. But for our purposes in this chapter, I want you to understand that the term disjunct refers to an organism's distribution that is not continuous. Okay, and you can see here that this map illustrates the distribution of three different species that are quite closely related. So in the southeast here, we have the gopher tortoise, which you won't see up in middle Mississippi, but down along the coast, they are there, the gopher tortoise. Okay, and then there are also two species in the desert Southwest. Here's probably a clearer example, and one that I would kind of rather you focus on for understanding disjunct distributions, because this is clearly a disjunct distribution within a species. So here in the reddish coloration, you can see the distribution of the boreal toad. So there's a picture on the left of a boreal toad, and it occurs throughout the northwestern United States. And you'll notice that there's a patch that is inhabited in central Colorado that is not continuous with the remainder of the distribution to the north and west of that Colorado patch. Okay, so this is a very clear instance of a disjunct distribution. And when you see this, I want you to be thinking about, are these two things the same species? I want you to think about all the boreal toads that live in Colorado and those that live in Utah, Nevada, Washington, Oregon, Idaho, etc. Okay? Are they exchanging alleles? Are individuals migrating between those regions? Okay? And at what point do we begin considering those different species? Those are all concepts that we've touched on in previous chapters. In the context of this chapter, I simply want you to understand that this is a disjunct distribution for a species, and given your background understanding of the influence of isolation on populations, you might realize that if this pattern, geographic pattern, persists for millennia, that we could end up with multiple species because they are isolated, they are evolving independently. Okay, all of this plays into the influence of geography on biodiversity. Because in order to understand the evolution of the biodiversity that we see today, we have to recognize that things like this have gone on in the past for millions of years and may be the reason we see lots of the species that we see today. So when we observe current patterns of geographic distribution, such as the current known distribution of tiger salamanders on the right, so it's up on the top right, you see a tiger salamander. Below it are the counties in the United States in which tiger salamanders have been recorded. Okay, so there's an interesting gap here in our area, but for the most part, they're throughout the central U.S. and kind of up the eastern seaboard. On the left, I want you to look at this map that reflects the likely distribution of glaciers over North America during the last ice age, about 25,000 years ago. And I want you to think about the ability of this ectothermic or cold-blooded salamander, this amphibian, to inhabit those northernmost areas that it currently lives in. So Wyoming, Minnesota, Michigan. Can you imagine that? Do you think these species were living in those areas at that time? Okay, clearly not, right? Amphibians do not live on glaciers. Not much does. And so I want you to think about what would have happened to the geographic distribution of this species during the Ice Age. Well, surely they wouldn't have lived in the northernmost areas that they currently live. And you know what? This map doesn't show how far down they go into Central America, but I would imagine as it got colder up north, it also cooled off in the south, such that areas that are too hot for them now were not too hot for them then. And they might have lived in additional areas. They might have expanded their distribution in the south, but certainly reduced their distribution in the north. So distributions, the range, the places that organisms live, change over time. And it's critically important that you understand this. And it's an important consideration of biologists studying the evolution of species, because we simply cannot assume that where things are today is where they've always been. This figure from your text is a map that shows the geographic distribution of European starlings, the species of bird on the bottom left. And so what's interesting is a number of these were released in Central Park in New York in 1896. Okay, just a few of them, right? But what's amazing is that by 1920, they'd covered the Northeast. By 1930, they were pretty much everywhere east of the Mississippi. And by 1950, they were everywhere except the very western coast. Okay, it's quite remarkable to see how they have expanded their range over time. This is an example of a species that was introduced. It is used to living in northern climates, right? It's a European species. So the climate here in North America is not unlike what it experiences in its native range. Okay, so if you see a small blackish bird, they're quite common in, say, Walmart parking lots and disturbed areas. You're not going to see this out in the middle of the forest. But if you see a black bird with a yellow beak, that's probably a starling. If it's got all these little yellow dots on its feathers that you can see close up, it's a European starling, a species that was introduced from Europe. Now, not all of these types of invasions are due to the actions of humans. Here we see a cow and a cattle egret. Okay, so you live in the Southeast. You have absolutely seen cattle egrets. So next time you're driving around the Southeast, anywhere where there are farm fields and water on the ground, if you live down along the coast, if you're driving I-10, you see cattle egrets everywhere. Okay, and the clearest way to differentiate them from native species is this kind of yellowish area on their head and on their back during breeding season. Okay, there are some native herons and egrets that are also white, but these are quite distinctive. They're quite commonly found in association with cattle out in open fields. Most of the native species you will only find next to a creek or a body of water. Okay, so why am I speaking about cattle egrets? The interesting thing about cattle egrets is that this is an African species. Okay, well, not so remarkable. We were just talking about a European species. What's most amazing about cattle egrets is that they showed up on their own from Africa in South America. Okay, so it appears that cattle egrets arrived in the New World on their own. Now, flying across the Atlantic Ocean in an airplane takes quite a long time. I can't imagine how long it takes for a bird. But one thing to think about is when you're watching this hurricane season, where all of those developing tropical storms come from, realize they all come from the African coast and come to the west towards South America, directly towards South America before they bend up and come towards Florida. So realize that weather patterns are consistent with bringing birds and other things from Africa to the New World. So this is an interesting example of dispersal, an extreme example of dispersal. Now, you might be thinking that we've already talked about dispersal, and we've also contrasted it with vicariance. Remember, vicariance is when we have one species that becomes two, they are isolated, they go through allopatric speciation. This is the separation of populations followed by differentiation. Okay, and an example that I've used before and is reiterated in this chapter in your text is that of the decapods, the small shrimp that live on either side of the Panamanian Isthmus. Okay, so Panama was the last bit of land that rose from the ocean to connect North and South America. It used to be the Atlantic and the Pacific Ocean were connected in this area. They no longer are. And when Panama rose up and severed that oceanic connection, it isolated populations on either side of that landmass. They have since speciated, but their shared ancestry is quite clear. And while examples like this are clear biogeographically and make the field of biogeography seem quite simple, there are very complex biogeographic histories that will require a great deal of study and focus in order to disentangle these evolutionary histories. Even a somewhat more complex but still quite simple example is presented by this species of plant in your text that has a circumpolar distribution. So it occurs all around the North Pole, basically. If you think of the fact that our globe is not flat, as illustrated in this map, but rather those continents wrap around a globe, those northern regions inhabited by this species are basically the entire coastline around that North Pole. Okay. What's interesting is that they also occur in mountainous regions in the western US, in Europe, okay, and also in Central Asia. So I want you to think about this in the context of remember the map of the North American glaciers I showed you for the tiger salamander slide? Do you think this plant that lives on rocks in cold areas, admittedly, do you think it lives directly on ice, right? Plants do not grow on ice or on glaciers. So during the last glacial maximum, most of these green areas along these northern coastlines that are inhabited by this plant now were 100% solid ice. And so the plant species could not have lived there. And just like the salamander, their distributions almost certainly moved south, where today they like it along these northern coastlines because it's some very cool temperature, okay? But when that was covered in ice, that very cool temperature could be found in southern Europe, in Central Asia, in central North America, okay? Now, once it warmed up again after the last glacial cycle, right now we are in an interglacial where it is much warmer, the species migrated back to these northern coastlines. And when I say plants migrate, to be clear, I'm not suggesting that a plant moves, right? It's simply that plants that live further south where it's too warm die out. plants that live on the northern edge of the range survive, reproduce, and their offspring, their seeds that are blown north, do well. Seeds that are blown south do poorly. Okay? And they continually expand their range to the north, chasing those ideal cool temperatures. But what this map illustrates is a really interesting phenomena whereby some populations have been left behind. And where were they left behind? They were left behind in areas that are climatically suitable. In Central Asia, they remain in the Himalayan region. High altitude, still cold. Okay? Right just to the north of Italy, in the Alps, high altitude, still cold enough for them. Okay? Now, in the lowland regions around, in France and Germany, not cold enough. They don't live there anymore. Okay? But they've hung on in those southern refuges. Okay? So it's an interesting concept that reveals how the changes in our Earth's, not just the geographic landscape, as we saw with the ratites, but in the climate, has affected the distribution of species. Now, the methods that biogeographers use to dig into and explore these histories of species that have these unusual or interesting distributions, they focus primarily on data available from the fossil record, but they also utilize relationships of living species because obviously it's those species that initiated the interest in the group in the first place. And so here's an example of a plant genus, Nothofagus, that lives in the southern hemisphere. So it's in South America, it's in New Zealand, it's in Australia. And it's long been wondered, why do they live on all these southern continents? I've shown you in that example with those ratites why organisms might live in those southern continents, because they were around way back when those continents were connected. Well, in reconstructing the phylogenetic relationships among members of this genus, okay, and then using phylogenetic dating methods that we've talked about a bit. Remember, I talked to you about counting and utilizing the number of mutational changes in order to calibrate how old a split is in a phylogeny. Well, using those types of techniques, biogeographers have been able to test the hypothesis that Nothofagus existed while these continents were all together, like the ratites, and now exist in all of those places because they were there before the continents broke up. Okay? And in this case, we can see that that is not consistent with the results that have been found. So the time scale on the x-axis applies to the phylogenetic tree, and you can see that most of those things, like the Fuscospora group, okay, they live in New Zealand, Australia, and South America. They are not more than 35 million years old, okay? And that is younger than New Zealand, Australia, and South America's connection, okay? Though it's New Zealand broke off a long time ago, okay? And so for New Zealand to be inhabited by a species due to vicariance, okay, the age of the lineage found in New Zealand would have to be at least 70 million years old, okay? Because New Zealand has been isolated for 70 million years. If it's not 70 million years old, then how did they get to New Zealand? If it's not vicariance, it has to be dispersal, all right? So remember, the surface of the planet did not look in the past like it does today. Continental drift has dramatically reshaped the arrangement of the continents that used to be in a much different configuration, as we spoke about on the second slide of this lecture. Here you can see a more northerly view where you can see the Laurasian continents as well as the Gondwanan continent. So Laurasia consists of Eurasia and North America, whereas the Gondwanan continents are those that we've already spoken of: South America, Africa, Australia, and Antarctica. Okay, and there's an interesting biogeographic pattern that's explored by the text in which the process of vicariance associated with separation of Laurasia and Gondwana was tested. Your text discusses an example of cypress, which I misspelled, trees that exhibit a history consistent with vicariance. Okay, so in the phylogeny of cypress trees here, you can see that there are two primary clades that they've highlighted, one colored in green mostly, the other in red. Note that the red clade occurs primarily on southern continents, South America and Oceania, like Australia and New Zealand. And the green clade occurs primarily in Laurasian continents. Think back to that previous slide, right? Laurasia is Asia and North America, okay? So these two clades are consistent phylogenetically in that they are monophyletic. All the things in Laurasia are monophyletic. All the things in southern Gondwana are monophyletic. And what really supports the idea that their distributions are the result of vicariance is not just that geographic correspondence with the monophyly of these clades, it is the fact that that common ancestor between the two in this divergence time analysis dates to approximately 160 million years old, let's say. And that timing is consistent with when Laurasia and Gondwana separated from one another. Okay? It's a very interesting proof of this idea that vicariance has influenced the geographic distribution of species on our planet through the process of continental drift. Another great example of this that you are probably more familiar with is the geographic distribution of marsupials on our planet. So these mammals that have their babies quite small, raise them in a pouch. Look at their geographic distribution on this amazing map on the bottom left, okay? They live in the Australasian region, well, primarily Oceania. And then they're also in South America and Southern North America. You're familiar with the possums that you see on the top left. Whereas when you think of Australia, you're thinking primarily of things like kangaroos and koala bears. Well, this is a remarkable geographic distribution if you look at the globe today, because these continents are so far separated and there are no marsupials in Africa or Asia. But if you look at the map on the top right, if you look at the arrangement of the continents during the time of Gondwana, you can see that South America, Africa, Antarctica, and Australia were all together. And what's interesting is the first things to break off of this were Africa and India, okay? And what you're left with is a string that is South America, Antarctica, and Australia. And so how would you, if you were a marsupial, well, first of all, I want you to think about where are there more marsupials, in the Americas or Australia? Well, I don't know how deep your biological knowledge is of marsupial diversity, but your intuition is right if you guess that Australia has greater diversity of marsupials. So if there are many more of them there, where do you think marsupials originated? South America or Australia? Okay, I would guess Australia. We don't necessarily know, or I don't know. But that has us having marsupials move from the Australian region to South America. And how would they do that? Well, it would be by crossing Antarctica. Now, that's an interesting thing to think about today because Antarctica is this completely inhospitable sheet of ice, right? But that was not the case in the time of Gondwana, okay? There are frozen, fossilized forests on Antarctica, okay? When the continent was in this configuration, it was not a landmass covered in ice. It was a forested, temperate region that was completely amenable to not only the distribution of marsupials, but also their existence there. Okay, so it's quite likely that organisms not only moved across Antarctica to South America, but they also lived there for extended periods of time. So now I've shown you some examples of vicariance, but it's become quite clear through a great deal of biogeographic study that because of the influence of extinction and the constant changing of climate and the geographic landscape, that dispersal has played an enormous role in the structuring of biodiversity across our planet's surface today. And this is apparent, no, this is uh, very apparent in the biodiversity that we find on islands. And some of the most remote islands in the world are those of the Hawaiian archipelago. And so clearly you can think about the islands of Hawaii and how the things that are there got there. Now, you may remember that we've spoken about Hawaii and that these islands are volcanic in origin. They did not break off of a landmass, so they simply popped up out of the middle of the ocean through volcanoes that broke the surface of the water because they got so large. Okay? The things that are there today are necessarily the product of dispersal. So when you think about isolated islands out in the middle of the ocean like Hawaii, think about the things that are there. We've talked about the Hawaiian honey creepers, the birds I've got up in the top left. But think about other things that are in Hawaii, okay? I haven't told you about many other species that exist in Hawaii. Would it surprise you to learn that there are other types of birds there? Probably not. How about flying insects? No, not so much. What about frogs? Do you think frogs have gotten to Hawaii? How would a frog get to Hawaii? Unlike a bird or an insect, they can't fly. Do frogs do great swimming across saltwater oceans? No. Okay. There are no native frogs to Hawaii. Okay. There are so many different groups that are not found in Hawaii because the way in which they would get there is so onerous that only things that are supremely adapted to dispersing are capable of reaching it and are found there today. So the biological patterns that we observe in places like Hawaii often derive great benefit from an understanding of the geological history. Okay, and Hawaii is a remarkable example of this. So your text speaks specifically about the islands of Hawaii. And one thing to think about is the actual age of the Hawaiian Islands. Okay, because as we think about Hawaii today, we think about these islands, that which of those do I want to fly to for my vacation, my honeymoon, whatever it is. Do I want to go Kauai, Oahu, Maui, or the Big Island? And you probably don't necessarily think about the fact that as you go from northwest to southeast, these islands get smaller. If you haven't been there and seen all of the islands, you wouldn't necessarily realize that as you go from northwest to southeast, the islands get taller. Okay, Kauai being the shortest island, Hawaii being the largest, tallest island. Okay, so they're taller and larger to the southeast, shorter and smaller to the northwest. It's interesting. Turns out that the northwestern islands are also much older, with Kauai being about 5 million years old and the island of Hawaii being less than 500,000 years old. So anywhere, let's say 400,000 years old. Okay. And the islands in between are intermediate ages, right? So Maui is on the order of 1 million years. Oahu is on the order of 2.5 million years until you get to Kauai, which is 5. Now, this is a remarkable example of the origin of islands. And this is a hotspot archipelago. And so what that means is there's a spot under the crust of the earth where things are pretty active, and it frequently punches through the crust of the earth, creating a volcano. Okay. Well, when it punches through and creates a volcano, it creates an island. The most recent island it has created is the island of Hawaii. Okay. The interesting thing is that the surface of the earth, the crust, moves, and it moves, in this case, in the Pacific, to the north and west. And you know what all these other dots are to the north and west of the really dark blue polygon at the bottom of your screen? Those are older islands. Nihoa, Necker, Laysan, Midway. Okay? These are islands that are not 1, 2, 3, 5 million years old. Okay? They are 10, 20, 30 million years old. Okay? But they have been around for so long. What happens to a volcano that's been exposed to the elements for 30 million years? It's rained on, it's eroded by rain and wave action, and they disappear. Many of these islands disappear beneath the surface, but they're still there under the surface where erosion no longer affects them. So this presents some really interesting hypotheses that could be tested by a biogeographer. If I were to imagine or think about the diversity of, let's say, fruit flies that live on the Hawaiian Islands, okay? And if I go there today, where would I expect to find the most species of fruit fly? Okay. Would it be on the biggest island or the oldest island? Okay. Well, it might depend on when they got there, but think about that bottom map that shows all of those extinct islands. How long do you think fruit flies have been in Hawaii? Can it be more than 5.1 million years? I don't see why not. It could be 10 million years, 20 million years. When they colonized the Hawaiian chain, what they colonized were, say, Laysan or Necker or Midway. And as Midway sunk, they colonized the next island in the chain. As Nihoa sunk, they colonized Kauai. Okay. And so the species that are present on the island chain today might actually be older than any of the current islands. And I would expect there to be a great deal of diversity on Kauai. And I would expect that Kauai may have been the source for colonization of these other islands. And when we look at a phylogeny, in this case not fruit flies, but a species or a group of plants that is native to the Hawaiian Islands, you can see that in this phylogeny of all of these species of plants, look at how the island of Oahu has been colonized. It has been colonized how many times? I would say 1, 2, 3, 4 times. Okay. Oahu has been colonized 4 times, those red lines. From 3 of those times, it has been colonized from a lineage, a common ancestor that was most likely from Kauai, right? One of the colonizations of Oahu appears to have been from the island of Maui, okay? So that's an interesting example. Let's follow this very topmost clade, where we go from the common ancestor being an orange lineage from Kauai that becomes red, colonizing Oahu, which then becomes green as we move up the screen, up the tree, colonizing Maui. And on Maui, we've got 2 lineages, Cajuana and Mauiensis. But their common ancestor appears to have colonized Oahu back. They flew back to Oahu, and 2 species originated there, and 2 more originated in Maui. A very complex scenario, but it is clear that the oldest common ancestor has a Kauai ancestry, okay? And they have colonized the younger islands in succession, consistent with our hypothesis. So now it is time for me to dig in a bit and think not, or at least allow you to think for a brief moment about the difference between the fields of biogeography and phylogeography. So we've been speaking primarily about biogeography, and this is exploring the relationships of organisms and the evolutionary history of different species with respect to geography. But we can apply these same principles to the study of patterns, evolutionary patterns within a species. Now, they're still considered a species, just like the boreal toad shown down here. But evolutionary analyses of genetic patterns within a species are clearly important and help us understand how speciation happens, how populations evolve, and how things specialize and adapt to new environments. So studying disjunct populations and understanding patterns of gene flow and migration between different populations are clearly helpful in understanding the evolution of species. Your text presents a really cool example of how exploring phylogenetic patterns within a species. So if I collect, a bunch of species of this plant, okay, this Symphonia globulifera, that occurs not only in Africa, but also in the New World tropics in Central and South America. If I collect a bunch of samples and look at how they are related to one another, much like I might do with my own genetic data in 23andMe, right? I can look and see how I'm related to other human beings on this planet using the same phylogenetic methods that you've learned in our previous lectures, okay? And in doing this for this species, Symphonia globulifera, the geographic area that is inhabited by these older lineages, the earlier branches on this phylogenetic tree, being from Cameroon and Dominica in the West Indies, whereas the more recently derived lineages are from South America east of the Andes, then successively west of the Andes and further north. Okay? Given that the outgroups are all African and Malagasy, that the earliest red lineage of globulifera is from Cameroon and West Africa, it would appear that we can look at this phylogeny and reconstruct the history of global movement of this species. It would appear that they came from Africa and colonized the Caribbean, Dominica being an island in the Caribbean, and then made it to South America, but on the eastern side of the Andes, then colonized, moved north and west, getting on the other side of the Andes and moving north into Costa Rica and Panama, but also moving to the western side of the Andes. Okay? And it's these types of patterns, especially when we explore them in multiple species that live in the same area, when we find similar or common patterns across species, as illustrated here in the phylogenetic relationships of populations within 2 species of fish and 1 species of plant. Okay? If you look on the left in Lipomus punctatus, you can see that all the populations from the Apalachicola River and the western panhandle of Florida, say Pensacola, Panama Beach, west, they all fall into a single clade. And those east of that, primarily those on the east coast, all fall into a separate clade. The same is true in Ammiacalva, another species of fish, and this plant species. So this split, this east-west split between these populations of each of these 3 species provides evidence that there was some biogeographic pattern, some force that isolated these populations and allowed them to become different over time. Now, you could drive 20 minutes between one location and another and catch a fish from a different evolutionary lineage without really recognizing that you had crossed some boundary. And these types of evidences are the ones that are used to reconstruct the influence of past events, such as those driven by glacial cycles that we've discussed, right? There are many species that during glacial times, when glaciers impinged on the northern United States, there are many species that like warm temperatures that might have only hung on in very southern Florida. So there are some really unique lineages of animals, specifically reptiles that I'm familiar with, that are found in Florida and nowhere else until you get over into Mexico. Okay? How did they get to Florida, right? They didn't just go there from Mexico, but they're likely only persisted in North America or in the United States in southern Florida after that glacial cycle. So, what are the things that influence the geographic range of species that prevent them from just being everywhere? Why isn't everything everywhere and we have all the same species everywhere you go? Well, we've talked about dispersal ability. And not all species have the same likelihood of moving or ability to move over distances. Remember, we've talked about the difference between dispersal ability of a lion or some big cat like a mountain lion and a snail, right? They clearly have very different dispersal abilities. We just spoke about the biodiversity of Hawaii, why certain things are there and others are not. Uh, Bats are the only mammals that are native to Hawaii. Why is that? Well, I would think that's quite obvious to you, right? So those things may limit the distributions of species. There are also ecological limits, right? So if you go into Central Africa and visit a beautiful rainforest and you go see gorillas, okay? These gorillas inhabit these rainforest regions of Central Africa, but why don't you find them further north in Egypt or in Morocco or southern Spain? It's because they can't extend their distribution beyond the rainforests that they currently inhabit and cross what is a very different biome in the Sahara Desert. Why don't we have elephants in South America, right? I spoke to you of an example, the cattle egrets, where cattle egrets came from Africa and colonized South America. Why didn't elephants do it? Why not lions? Okay. Well, clearly, these are organisms that have different capabilities for dispersal and different ecologies. Okay. And this is something critical when we think about the geographic distributions in the evolutionary history of species. Now, one of the things that your text highlights is one that I wouldn't normally highlight because I'm not a plant person, but this is a fantastic example of the influence of mating style of plants on their ability to colonize different areas. And so some plants are able to self-fertilize, right? And that makes things quite easy. How many plants do you need? How many individual plants? How many seeds do you need to create a new population on an island if that species of plant is capable of self-fertilization? If they can self-fertilize, you need only a single seed to wash up on that beach, grow, it can self-fertilize, release more seeds on that island, you get a whole population just from one seed. If this species requires outcrossing, so some plants are unisexual, right? They have a single sex. They are either male or they are female. They must outcross. They must breed with another individual. Well, that, of course, reduces their ability to expand their distribution rapidly. And this figure in your text indicates the relative range size of selfing species versus outcross species. So each bar represents a group of plants that are closely related to one another. And how big is the geographic range of those individual species within that clade that can self-fertilize versus those that require outcrossing? And you can see here what you take from this is in all but one of these groups of plants that were studied, the self-fertilizing species have a larger range size. Okay? So what about the interaction among species? How might that limit geographic distributions and range sizes? Well, what's interesting is we've spoken and learned about allopatric speciation. Two populations become isolated, they diverge from one another and become different species. But what if conditions change, bringing those two populations back together into the same place? Can they coexist? Well, that depends entirely on how much they have diverged. Okay? So, in two different important aspects. One, did they diverge enough to be reproductively isolated? Will they no longer intermate, intermingle, interact, exchange alleles, and fuse back into one species? If they're not going to do that, okay, then it really boils down to, are they fighting over the same resources? Okay? So on the left, you can see a pair of taxa that are very similar to one another, utilize very similar food resources, and have a very hard time living in the same place because they're just competing with one another directly. On the right are two closely related species that have diverged in they live in the same habitat now. They have diverged in the way in which they secure their food and the specific prey items that they go after. Okay? And so you can see that in this graph in between them, this amount of divergence. If there is more divergence, how long does it take before they can be sympatric? Well, if the divergence is great, very high, say 0.6, then you don't need much time before these two populations can be sympatric. But if divergence is very low, there's going to need to be a long period of separation before they are able to live sympatrically. So the ecology of individual species affects their geographic distributions and their ability to expand and interact with new environments. And this is true of species that are introduced. Okay. In the United States here, we have many, many species that have been introduced. Okay. One of the ones you're probably most familiar with here in Mississippi is kudzu, the plant that grows and covers everything in the summertime. That's a species of plant that was introduced from Asia. And if you go far north, if you go up to Michigan or southern Canada, you're not going to find kudzu because it doesn't live in those types of environments back in Asia, nor does it live in those environments here in the United States. It thrives in hot, moist environments. Well, the same is true in this figure. It's illustrating the relationship of where a number of reptile and amphibian species live in their native range on the x-axis, primarily focusing on how far north do they go in their native range, and comparing that to the geographic distribution, the northernmost range limit of the introduced populations. And the fact that this line is nearly a straight one-to-one slope of one line suggests that the northern range limits of where they came from pretty much limit where they can go in their introduced range. So, for example, iguanas have been introduced into southern Florida. They're incredibly abundant. They're all over the place. But you might have seen this winter when we had this incredible freeze that came through, that there were a lot of news stories of falling iguanas, right? Because iguanas are not made to tolerate, they're not adapted to tolerate freezing temperatures. It very rarely freezes in southern Florida, in the Keys, in Miami. It only happens, once every 10 years or so that they get a good hard freeze. And when they do, these iguanas fall from the trees, okay? They simply cannot handle those low temperatures. Now, there's one concept that we haven't yet touched upon, and that is the limitations of organisms' ability to adapt to new environments. So simply because it would be beneficial to adapt a new mutation or niche for a particular species doesn't necessarily mean that's going to happen, and that's something that we've covered already. But what I want you to understand here is that there can be a phylogenetic component to those constraints, and the term that we use for that is phylogenetic niche conservatism. And what this means is that the geographic distribution of some species may be limited by their ability to adapt to the environments that are in the areas around them. So, for example, an organism isn't going to disperse across a completely inhospitable region. I gave you the gorilla and desert example. Organisms aren't necessarily going to adapt to very, very different environments simply because it would be beneficial for them and allow them to expand their distributions. Under phylogenetic niche conservatism, species have limited ability to adapt to different environments. And you can see here on the left the concept illustrated whereby there are 3 clades, each containing 3 species. And within each clade, each of the 3 member species has a very similar ecological niche, the niche being represented by these different sized and colored lollipop shapes. Okay? So, for example, the ones on the left are adapted to, let's say these are tall trees as opposed to medium-sized trees or very short trees, right? And within each clade, there's no variation. The phylogenetic history of those three species constrains them and does not allow them to adapt to live on one of the other environments or use the other niche. The opposite of this is illustrated in the tree on the right. What you're seeing there is not phylogenetic niche conservatism. The niche is not constrained by evolutionary history. Within each of these three clades, you can see that each individual species has a very different niche than the other two. Okay, so this is a concept that's reviewed in the text, and I encourage you to review it as well. So the last bit that we're going to touch on in this chapter hits upon global patterns of biodiversity. And just a very simple concept that you're almost certainly already aware of is the abundance of diversity in equatorial regions. When you think of areas that have lots and lots of biodiversity, what probably comes to mind is a tropical rainforest. Well, where do you find tropical rainforests? Or maybe it's a coral reef. Where do you find coral reefs? All of those are found in equatorial regions. Okay? As you get further north from the equator or further south from the equator, the temperature is not so stable. Okay? In the equatorial regions, the temperature is very stable year round. And that stability can be beneficial to the accumulation of biodiversity. If you have glacial cycles, as I've described already, there were glaciers covering part of North America. Do you think there were glaciers in the middle of Brazil near the equator? Certainly not. And so this stability over time, this area, the incredible productivity, there are many more species of plants, meaning there are many more species of insects, meaning there are many more species of animals, other animals. This all contributes to this dominance of the equatorial regions in terms of number of species. This figure from your text simply illustrates the number of vascular plant species per 10,000 square kilometers. And you can see that as we go from the left on the x-axis, which is the equator, to the right, which is moving further and further north, you can see that the number of species decreases pretty dramatically. All right. So this boils down to what has really, or the question, I suppose, is what has really driven this higher biodiversity that we see in these tropical or equatorial regions. And on the right are three hypotheses that have been proposed that would explain why tropical diversity is higher than temperate diversity. So tropical regions, you're familiar with that term. Temperate refers to regions such as Mississippi or Michigan, where we have distinct seasons. You have a fall and a winter, you have a spring and a summer. Okay? These three hypotheses attempt to describe why this green line is higher than the blue line, in short. The green line being the diversity of species in tropical regions, the blue line, the diversity of species in temperate regions. You can see the x-axis is time. So why is it that over time, the number of species in the tropics has exceeded the number in temperate regions? And I've bulleted them here. Is it because there's either a higher K, a higher S, or a longer T? Okay, that's what these three graphs really boil down to. And we've discussed what K and S are, right? Carrying capacity and speciation rate or diversification rate. And so you can see in A and B how what is being illustrated here is that in A, a higher carrying capacity. With your understanding of carrying capacity, you should now understand what is being illustrated in figure A. They both started out the same, the trajectory was the same, but temperate regions leveled off earlier because they simply cannot support the number of species that tropical regions can. In figure B, they started out at the same point, but the green line has increased faster because the speciation rate is higher. And then the longer T, that simply suggests that species have been evolving and diversifying in tropical regions longer than they have in temperate regions. And that might not make sense when you think about the context of the planet being a single age, but I want you to think about how long have frogs had to diversify in Brazil versus how long have frogs had to diversify in New York, given what you saw happened during the last glacial cycle. Okay. And so that is kind of the context for this uh, time consideration in the accumulation of biodiversity. Uh, on the left here, there's a phylogeny of frogs showing a bunch of lineages, how old those lineages are, and they're color coded at the tips by where those species are. Okay. And you can see that there are many more species in tropical regions than there are in temperate regions. But what's interesting is that the lineages are incredibly old and they have been in these regions for a long time. Okay. So it appears to be that time has played a significant role in the accumulation of frog diversity. Okay. But I encourage you to read this section of the text to ensure that you understand this. Okay, as this is the last week of the semester, I'm not adding additional reading for this week. So this will conclude the material that you will be responsible for on your final exam.